when it comes to drugs, vaccines, new products, they have to go through a process of development before they get onto the market. There are actually a lot of drugs that are being tested here. When I first started out in my career, it was strictly infectious disease. Even when I was working when I was actually practicing medicine, most of the cases that we used to see, a lot of the disease burden was infectious. Research has a bad history of preying on the weak. And that's not the only way. There are studies where people went to hospitals and they looked on mentally retarded children. For us as a nation, because we don't have a muscle to do the whole value chain, we can just pick a small part and do the innovation part, do the part that we can. So ethylene glycol is actually used in the automotive industry, and it is actually very toxic, but it's very sweet. So it was used to formulate this medication and given to children. And there were a lot of deaths and there was a lot of hospitalizations as a result of that. My name is Grace Kaguthi. I am a physician and clinical trialist. That means I trained at the University of Nairobi in medicine and surgery and graduated in the class of 2006. Thereafter, I did my master's in clinical trials at the University of London and then proceeded to complete my PhD at Erasmus University in Rotterdam in the Netherlands. So since that time, I've really been working a lot in clinical research for more than 15 years. And I also have an advanced diploma in pharmaceutical medicine. And I'm about to complete a Master's of Science in Neuroscience and Psychology at the King's College London.
Would you probably explain why it is necessary to acquire all these qualifications?
Yeah, for some of them, they were essential in my line of work. So, for example, obtaining the Master of Science in Clinical Trials was necessary because I'm working in clinical research, conducting clinical trials. So it was a natural fit. And the PhD, while not everybody gets a PhD, for me, I found that it built up my skills and my critical thinking in a way that is necessary. The advanced diploma in pharmaceutical medicine has some industry-specific aspects that you don't get from all these other training that I did. And then the last master's is just because I like to read. Okay. So it was just curiosity.
All right.
Yeah.
Earlier on, you described yourself as a clinical trialist.
Right.
What is that?
Okay, so when it comes to drugs, vaccines, new products, they have to go through a process of development before they get onto the market. So if you think about vaccines, COVID vaccines, or antihypertensive drugs, anti-glycemic drugs, any drugs of any kind, before they actually end up at the pharmacy where you get them, they have to go through a process of testing. And there's usually a preclinical phase where they're tested in labs and in animals, and then they proceed to a clinical phase where they're tested in humans. So the clinical trialist is the one who designs the studies that are going to be conducted in humans, analyzes them, conducts them, reports them, and then they are usually submitted to regulatory authorities to make the decision as to whether they are safe and whether they are working, whether they are efficacious enough to actually go to a pharmacy. So the clinical trialist has custody of that last process in humans, and it's in phases, but I may not get into that right now. But the clinical trialist is the one who participates or directs that process. In the early days, like in the West, people actually just used to make drugs in their labs and formulate them in a way that is palatable to the public and sell them. So there was actually incidences where that turned out to be a disaster. There was actually a drug that somebody formulated that was being used in pediatric populations that was formulated with something called ethylene glycol. So ethylene glycol is actually used in the automotive industry, and it is actually very toxic, but it's very sweet. So it was used to formulate this medication and given to children. And there were a lot of deaths, and there was a lot of hospitalizations as a result of that. There's also been other disasters, like in the 50s, there was a medication, an anti-nausea medication for pregnancy in the first trimester. So, women tend to get morning sickness. And so this drug was marketed. Someone developed it in the lab and discovered actually can help with morning sickness. And once it was given to pregnant women, they started to observe what they call phocomelia, so children who are born without limbs. And it turns out that thalidomide, this medication, actually has a mechanism that is interfering with the development of limbs in the womb of the fetus. So the process of testing drugs and to make sure that they are as safe as possible, or rather that the benefits outweigh the risks of using them, is critical. It's a very important part of the medicines that we use to make sure, to the extent that we know that they are safe and they work. And without that rigorous process, which actually takes a very long time, we don't have confidence to use these medications. And I think that's some of the challenges that come with herbal medications, because they haven't been through that rigorous process of finding out what's the right dose, who should take this medication and who shouldn't, because everything has a benefit and a risk to it. So that's why this rigorous process is necessary. There are actually a lot of drugs that are being tested here. When I first started out in my career, it was strictly infectious disease. Even when I was working when I was actually practicing medicine, most of the cases that we used to see, a lot of the disease burden was infectious. So there's a lot of HIV and all that. The antiretroviral drugs had not yet been scaled up. But with time, we've actually seen an evolution where there's more sophisticated research taking place. So it's actually become more diversified and more specialized. As a field, the research has become more diversified and more specialized. So even clinical trials was not a master's that was there maybe 10 years before I did it. It is something that has evolved and become necessary with time. And I think just because the industry is not so mature here doesn't limit anyone from taking such a course, because once you take the course, then you're equipped to work in that industry and to build it, which is where we would like to see Kenya and the rest of the African continent go, to become more sophisticated in developing our own research methods and our own research industry, even our own pharmaceutical industry. It's a process. And maybe I can use this opportunity to highlight Reddy Pharmaceuticals in India. So Reddy Pharmaceuticals in India, they realized that one thing that hampers the development of the pharmaceutical industry and its maturity is the issue of patents. So you need to file a patent for every discovery, and there's a specific criteria. It's not something we talk about a lot in Kenya, but intellectual property rights are important. So Reddy, they took away the issue of patents and allowed their pharmaceutical industry to develop generics. So the process of developing an original new molecular entity is complicated. It takes time. You need certain labs, you need certain chemists, and you need a certain group of specialists. So they took away that and allowed their pharmaceutical industry to copy. And in copying, they began to learn the trade of the tool, how to formulate drugs, and then they developed certain strengths, like I'm telling you, in formulation, I may not be able to develop the drug from scratch, but I can create a better formulation. I can create, instead of you swallowing a tablet, you can inhale. You can use an inhaler, for example, for asthma. So that is how they grew their pharmaceutical industry. They didn't start from scratch. And sometimes that is what has worked to their advantage, because sometimes you're so far behind, it will take you quite some time to catch up with where the rest of the world is. So they did that. And the rest of the world was not happy, of course, because if you have a patent, you spend time, this is your intellectual property, but they scrapped it. And then after that, they used that platform of copying to build their manufacturing, their formulation, and actually now they've ended up having their own kind of patents and partnerships with the companies like in the US, and their manufacturing is actually recognized by the FDA. So the clinical trials, they are still relevant. It may not look relevant now, but the world is growing, the country is growing, the region is growing. So those skills are actually increasingly going to be needed.
I'm getting the impression that what happened in India was a deliberate disregard of the international intellectual property regime. They just said, forget about it.
Right.
Is that a correct assessment? And
that is 100% accurate. That is, they completely flouted those rules and disregarded the international trade laws, and that's what they did.
So, in your own estimation, in the coming to Africa, does the formula for the rejuvenation of pharmaceutical industry lie in disregarding international IP regime?
I would not go down that route just because they managed to do it and got away with it. I think right now it would be difficult for anybody to do that. But I think as a people, we have not sat down even to ask that question, to ask how can we build our own research industry, even though we financially we may not have the might to do things that way. But that actually is an opportunity, because that's your opportunity to innovate and come up with a way to build things that are not from scratch. Right now, there are technologies that are there now that were not there before, that are accessible to us, like AI, artificial intelligence. There are new ways of doing things that don't require us to have everything that the other countries had when they were starting out. Secondly, we can also learn from their mistakes, like I was telling you about thalidomide and the ethylene glycol scandal. So there are ways we can have shortcuts without necessarily flouting the rules. But the first place is to ask the question, what about us? What are we doing to get our research industry off the ground, to make our own drugs, to make our own vaccines, everything? Actually, most companies who don't have the financial might of the big names that are listed, for example, on the New York Stock Exchange, the big pharmaceutical companies, that's what they usually do. So you start from making generics. So they watch that patent timing. From the time you file the patent to the time it expires is 20 years. And that's really just about most companies really work hard to just fit into that time limit and make sure they conduct all the studies and just before the patent expires. And they usually don't end up having a lot of time to sell their medications. So many generic companies in the US, in India, are watching that patent expiry like hawks. And the moment it expires, they are going to flood the market with generics. And generics, you can only sell them for 6 months exclusively. But it still makes a difference. So it's an excellent starting point. And I'm glad you brought up that point.
You said earlier on that it takes about 20 years for a patent to be registered.
To be valid. After that, it expires.
Yes.
Yeah.
Okay. So basically what you're saying is that from research all the way to development and commercialization, it takes a whole lot of time.
Right.
But we also saw, maybe you could explain, at least how that process works.
Yeah.
Because I don't know how to ask you about what happened during the COVID vaccine development.
Okay.
Because it looks like that period was compressed. But explain the process of developing a drug.
Okay, excellent. So usually, like I mentioned, there's usually a preclinical phase and a clinical phase. So we start from discovery. So even in discovery, things have changed so much. Before, people had to do a lot of experimentation and, let me try this and let me try this. And the discovery was like serendipitous. But right now, companies have libraries of chemicals. So they just develop a series of chemicals. And of course, they're tapping into intelligence systems, artificial intelligence systems, and so on to create these libraries. So you discover this drug, and you discover that it can work for heart failure, or it can work for multiple sclerosis, whatever the issue is. So once you discover it, there are certain in vitro tests that you have to do. In vitro means you do it in the lab or in silico. There are certain, you can use even modeling, because these days the knowledge has advanced to the point where we know that if we see this kind of pattern or this kind of moiety, we can predict that this one will cause liver toxicity. We can predict that this will cause thyroid toxicity. So you eliminate those molecules or you tweak them. So that's the in vitro stroke in silico phase. You do it on a petri dish, whatever it is. Then from there you move on to animal studies. And the animal studies are mainly toxicology studies. And in that time you want to find out, if I give this drug, does it mutate Is it mutagenic? Is it going to change the human DNA? So those are tests of mutagenicity. And just give me time to explain, because people need to understand that these drugs don't just end up on the shelf by chance. So you do those tests for mutagenicity. Then you also do tests for carcinogenicity. So you do these drugs induce cancer. And how you do that is you get these animals, maybe mice or guinea pigs, even monkeys, and you inject them with toxic doses. And it's a balance, because you give too high overdose, you kill the animal. So you just need enough where you can cause it to be tumorigenic, to cause tumors to form. And then you need time for those tumors to develop. So these carcinogenicity studies typically take maybe 5 years or so. It will take probably the lifespan of the animal. So that's already chewing time into your patent. So that's carcinogenicity. Then there's what they call safety pharmacology. So you look at key organs. You look at the heart, the brain, and the spinal cord, which we call the central nervous system. And then you look at the respiratory system, the vital organs. Are there effects there? So you administer to these animals, and then you observe them. What's their respiratory rate, what's their heart rate, and so on and so forth. Then there's also other things, like things that we didn't know before, like addictive potential. So I don't know if you remember, but there was an opioid crisis in the US, because there was a company that sold these drugs to people knowing that they were addictive, so that they could keep buying and taking. So you have to, these days, you need to test these drugs, whether they have addictive potential. So basically you put this rat or the mouse in this cage, and you see how much you allow it to self-consume. So it can roll on a track wheel and feed itself on that thing. Then you know whether it has some addictive potential. So you do all this battery of tests, and that's going to take you maybe 2 to 3 years, and so on. Then now from there, you need to determine the safe starting dose to do a first in human study. So now you've done in lab, in animals, now you go to the human phase. So the phase 1 study also takes about 2 to 3 years. You look for healthy volunteers, and then you give them this starting dose. You want to make sure that their organs are functioning well, so that should there be a risk, they can be able to bounce back. So you don't want to give it to somebody whose organ function is compromised in some way, because they're not able to bounce back. So strictly healthy, except for very toxic drugs like chemotherapy. There's no, ethical justification to give a healthy person a toxic drug like chemotherapy. So that's phase 1. Once you're done and you found that the drug is safe in those very small numbers, then you go to phase 2. Phase 2, typically you test it among people who have the disease. So if you have an antihypertensive drug, you want to find people who are hypertensive. And ethically, it's because we know the antihypertensive treatments, it's not allowed for you to, usually we do in 2 groups. We have a test group and a control group. But because we know that the antihypertensive treatments already, we can't deprive one group of antihypertensive treatment. And this is a disease that is fatal over time. So they will be on the standard of care, what they call standard of care. So whatever treatments are there, they will get. Then in the test group, now you add your experimental treatment. That's phase 2. Now, if the treatment works there, and many drugs and vaccines and biologics tend to fail there, then you can move on to phase 3. These are called the confirmatory studies. And these ones you do in thousands or hundreds, much larger numbers, and usually typically people with the disease. So already you can, if you are counting the years, you're probably, by the time you're in phase 3, you're probably like 12 to 15 years in. So you only have 5 years left on the patent. Once you're done with the phase 3 study, there's a process called filing. So you submit it to a regulatory authority. So they look at your data and they determine whether you conducted the study in a way that is credible. Like, can we really believe this data? Is it robust enough? And can we trust that this medication works? And that process can take like another year or so. Then now you hit the ground running to launch the drug and try and extract as much as you can from that process. Now, that whole process, I didn't tell you, not only does it take almost 20 years, it can also, it costs, this nowadays, the costs have really gone up. So it costs about $2 to $4 billion from discovery to market. So you can see it's a very costly process. So for a country like ours, where we don't have that kind of muscle, financial muscle, and generally, most countries, it's only at the top, at the phase 3 point, where you look for these big bucks. In the other stages, you're working with different institutions. So universities tend to do the discovery, and then other partners can do the early phase studies like the biotechs, and then they sell these molecules, and like that's what happened like with the COVID vaccines. They had been developed by small biotechs. Moderna was a small biotech, and then it sold its product to, I mean, it listed on the stock exchange or mobilized funds in some way. Same thing to Pfizer. They licensed out that technology and bought it from a smaller company. So there are many people involved, but all this trading is expensive. All this development is expensive. And that's why I'm explaining this process to show that we are behind.
It raises the question of if it takes such a long time and costs so much money,
right.
It means we may not be able to afford it, or maybe, to put it differently, we are not doing it.
So one approach, which is what happens a lot, is if like we were talking about Facebook and about Microsoft. So the people who develop the technology or the discovery of these drugs is not the people who commercialize it. So most cases you find the discovery is not happening in the big companies, just because the company is not set up to discover. The company is set up for different things. But discovery happens in universities, in these small biotechs, just exactly what happens in other industries. If somebody is going to come up with an innovation, like even here, like M-Pesa, I understand it was a university student. So innovation is happening very different from the space of commercialization. It took Safaricom's infrastructure, muscle, networks, knowledge, expertise to scale M-Pesa and to keep it running on the level that it's running. But the discovery is happening at a very small level.
I was going to ask about the research ecosystem.
Okay.
In this country.
Correct.
What is the status of research in universities, in the so-called, what you're calling small labs,
yes,
and organizations?
Right.
Do we have any kind of research going on there, or and if so, what kind of research? What's promising?
Right.
What's not? What's the status like?
So just before I get to the status, I'll get to it. So I was just saying that for us as a nation, because we don't have a muscle to do the whole value chain, we can just pick a small part and do the innovation part, do the part that we can. We may not have the billions of dollars, but we can certainly innovate in our university. There's a lot of innovation that goes on. That in discussions, in meetings, people come up with ideas that don't see the light of day because we lack a proper ecosystem. For example, the patents. The patents are very important in protecting your intellectual property, because how will you reap the reward of your labor? So that research ecosystem which you're asking me about is kind of fragmented. It's not mature, and it's something we need to think about building it. So when we have innovation in the universities, usually, like, people don't have enough information about, like, universities should probably invest in training students on intellectual property. I didn't properly understand patents and intellectual property until I started working. That's a huge disadvantage. They should have invested in teaching us about intellectual property, whether it's copyrights, whatever, music, whatever things that you come up with, you should protect them with a patent, because that allows you to commercialize and reap the benefits. You need to be rewarded for that effort. So that's number one. We need a patent system that protects somebody's intellectual property. Then the other part is we need a way of connecting that innovation to somewhere where it can be refined and where it can be explored further. So you need a financing mechanism, and governments in other countries tend to finance universities. And then universities it's a cycle. So the universities now end up generating these new innovations that end up making money and paying taxes. So there's a virtuous cycle in place that we need to set up here. Maybe we might need a bit more government funding, but even apart from governments, there's a lot of non-governmental funding that goes to universities from philanthropists in other countries. And this country has a lot of wealthy people. So they can consider, because health affects everybody. Cancer affects anybody, everybody. They should also consider funding these small biotechs and in universities. And there are structures they can put in place to make sure that there's fiscal discipline to fund those small biotechs and universities. And then institutions like KEMRI, which are like the government mandated medical research institute, should now partner with those students. And there's a lot of research that goes on in KEFIS, for example, the Plant Health Inspectorate, CalRO, Agriculture and Medical Research. But the funding is too small and fragmented. So it's the full circle, the full value chain is not happening because we don't have the big bucks, but we can do this small part. And then now we can license out these products. And there's a lot of money to be made in licensing out these products that can keep this research ecosystem going and build it up to a place where actually we can have a serious pharmaceutical industry if we are patient. Because research is like you have a field. I'm sure you know the story. You can have a field and a treasure in the field. But then it's not the whole field that has the treasure. So you have to buy the whole field just to get that little treasure. So one thing I didn't mention was when you're developing these drugs, 90% of them are going to fail. So you have to buy the field, but for that treasure you get, you can extract enough out of it to sustain the industry.
Going back to research funding, everything really depends on basic research.
Right.
So at what point are we really failing? Is it in the funding of basic research or in the funding of refinement, research and development?
Yeah.
Where are we really failing?
Well, we're not strong on any of those fronts because basic research requires a certain level of investment in labs, in human resource. So we have not done much. We have an elementary level, but we need to do much more to grow our basic research. And usually how people do that is by applying for grants, being attached to senior colleagues, professors who have the name to attract the funding. But sometimes even the senior professors, they may not hold it in their consciousness that they need to build a system to bring up the young ones, to allow their innovations to thrive. So it's a bit of a pyramid with one person at the top. So that needs to be disrupted also by changing our thinking.
I was going to ask you, now that you have said that we are not doing very well in terms of research, is it out of ignorance of the potential for research? Because as you said earlier on, it's possible for this country to become wealthy on just a single discovery.
Right.
So do we really know that? And what's the problem? Why are we not doing enough research that is homegrown?
So that has many aspects. The first one is we tend to have a consumer mindset when it comes to research. So we are more than happy to receive drugs that have been tested outside and just adopt them here. Like just guillotine, we use them. So we need to shift from our mindset from being consumers of research to being producers, and that's where the payoff is. So far, like for me, most of my career has been built on working on TB, malaria, sickle cell disease, and these are externally funded. So that's a disadvantage because like for us now, we realize that there's a dual burden of both infectious diseases and non-communicable diseases. But if you look, most of the research is in respect to infectious diseases because that's what is being externally funded. So we have not really sat down to think about how we can fund our own research based on our own disease burdens here. So if cancer is a problem in the West and they're doing those studies, then we'll be consumers of the research also, and then consumers of the drugs later. But we have not thought about promoting ourselves and doing our own research. And it's not just doing our own research. Like I said, we need to start training people in the universities to think a certain way. We need our schooling system to cultivate a sense of curiosity. We need our media to shift the discourse from politics and bad news to issues. How do we solve a problem? This cancer is a big problem here, specifically this type of cancer. Can we have some people come and discuss with us what the problem is and how we can solve it at a high level and on a continuous basis? So that education also has not happened. and there are some myths around that we can discuss. But essentially, we have not set up our thinking to solve our own problems. We tend to just hit and run. So as a problem is coming, I solve it. But I don't step back to think of creating a system that serves everybody.
Okay. Now the issue of your kind of work comes up.
Okay.
And from what I heard you say, it's like you are really concerned with the protection of people, the citizens.
Right.
In other words, what you do is ultimately geared towards protecting us.
Right.
But we know that there are sometimes nefarious forces,
right,
involved in research. Case in point being the Kaguthi, experiment, and there could be others. The drugs that we have in this country, that we import into this country, do we have a mechanism of doing local tests to ascertain their safety, or do we just assume that because they have been approved by other bodies in other countries, then they must be safe for us to consume?
Okay. Yeah, so you've actually asked a two-pronged question. The first part is about when we are investigating these drugs, and then the second part is when they are approved in other countries, how do they end up here? So the first part is, like you were saying about the Kaguthi study, so I don't know whether your audience knows about it, but essentially African Americans who had syphilis, even when treatment for syphilis became available, they were not given that treatment, penicillins and other antibiotics, when they became available, just so that, for research purposes, just so that they could observe the natural history of the disease, which is quite nefarious if you think about it. And there's been many such incidences. Research has a bad history of preying on the weak, and that's not the only way. There are studies where people went to hospitals and they looked on mentally retarded children and injected them with all kinds of things. During the Nazi regime, they also did all kinds of inhumane experiments that did not really have a proper scientific rationale. So that's an unfortunate part of research. But now research is very highly regulated and there are eyes watching all the time. So it's very difficult for you to just arrive in a remote part of Kenya and inject people or, administer something and disappear. Research has become very highly regulated, and in Kenya there is the Pharmacy and the Poisons Board, which actually has an expert committee on clinical trials that appraises all the research. They look at, if you want to do a study in Kenya, you have to submit your research to them for approval, and they have the right, even after they've approved your study, to monitor, to inspect. And there are also ethical committees which review the ethical aspects of your study. Is this study beneficial? Is it preying on the weak? Are you overly incentivizing people? Like, are you paying people so much money that they can't resist being in your study without fully considering what it means for them? So these research regulation mechanisms are in place, and they're relatively robust. They're not perfect. We can still grow, we can do better. But there is some mechanism in place. I would like to assure everybody that it's hard to just arrive here and do whatever you want. And likewise with new medications, even the COVID vaccines, after they were approved, they came here. There's a lot of batch testing. There are a lot of documents to be submitted to with regard to how they were manufactured, to make sure that their quality is acceptable, that they are safe. Because sometimes you can have a good product, but your manufacturing process introduced impurities, errors, maybe the cold chain in how it was transported from where it was manufactured. Now there's a rigorous process at that. The same Pharmacy and Poisons Board, but a different committee, is going to look at those aspects of quality and safety. And that happens in many places in the world.
Explain batch testing. Do they, for instance, go to the field and grab some of the tablets, as it were?
So the applicant, either the company that is trying to do that is going to have to provide samples for testing. And there's also certain documentation, and there are different aspects that are looked at. Like I mentioned, one of them is impurities, and then there's, I mentioned also the temperature regulation, because when drugs are not kept at the right temperature, you can't assure their quality, you can't assure that they're going to work as they were designed. So those samples are tested. And even once you actually finally ship your whole consignment here to be sold, there's also some testing that happens to make sure that those drugs are of the right quality.
You mentioned COVID, and the issue there was that the vaccines that we got were approved in a hurry, particularly those of the, were you calling them mRNA?
mRNA, yeah.
Yes, yeah. So what facilitated that kind of hurried approvals?
Yeah. So, yeah, it's true we had talked about the process takes up to 20 years or it can take a long time. The reason that happened was, like I mentioned, research is an ecosystem. So those technologies had already been in development for many years. From the 80s, the 70s, there was already talk of how can we administer drugs or vaccines using this platform called mRNA. And so it had been, that time it was being used to test as a mechanism to treat cancer. So all that was needed to do was transfer that technology, not now for cancer, but as a vehicle for COVID vaccines. So that already collapsed a lot of preclinical testing because that work had already been ongoing. That's the first thing. And then another thing that makes research take so long is this research approvals. So for example, if you submit your protocol to a Pharmacy on Poisons Board or any other regulator in the world, it takes them some months maybe to review the research. But in light of the pandemic, that everything was shut down, everybody was expediting their review. So their review times were shortened. Everything was shortened in light of the urgency. Another thing that makes these things take long is the studies. So it takes time to recruit the people into the studies. But for these COVID vaccines, the lines were going down the street. Everybody wanted to get a COVID vaccine because it might protect them. And all that abbreviated the timelines. Now that doesn't mean that there was not enough rigor in making sure that they were safe. That was still there, and that's why what they got is not necessarily a license. Initially they got what is called an emergency use authorization, and that's different from a license. A license, you need the bar is higher, you need more evidence of longer term safety. But emergency use authorization is something that, for example, the FDA did to expedite the provision of an intervention that could probably save lives. I don't know if you remember the images on TV. There were mass graves in some parts of the world. It was terrible. So it was in light of that they have provision to do what is called emergency use authorization, and then with more data you do what is called licensure.
There has been a question around technology. The old technology vis-a-vis the new technology. mRNA is a new technology.
Right.
And part of what I've read is that by its very nature it's synthetic. I don't know what that means, but I'm happy to use the term. But based on what I've been able to read, they say that these synthetic technologies can actually allow for programming. I think they have a distinction between mRNA and mRNAi, whatever that is. And that is the crux of the matter, that these technologies are programmable.
Yeah.
They can be programmed to work in certain ways based on genes and demographics and stuff like that. So do we have the capacity as Africans to verify that kind of
mRNA? In brief, what happens is in your cells there is a blueprint which we call DNA, and it is from that DNA that you generate all your features, any proteins, your eye color, your hair color, your skin color, everything's coming from your DNA. Now usually the system is so intelligent that it doesn't copy directly from that. So it makes a copy of your DNA and uses that copy to make the proteins and your features and whatever your body needs. So mRNA is usually what is used to translate what from the copy to the manufacturing plant in your cell. So if I give you an mRNA vaccine, your DNA is intact. I have not touched it. I have just piggybacked on a mechanism that transfers that information from the copy to the manufacturing plant of the cell. So it is impossible to program because it is not getting into the DNA. And I don't know why that misconception has really gained a lot of traction. It is patently false. It is impossible. You cannot hijack that system because it doesn't work that way. With any vaccine, once you inject it, typically, you're going to have what they call local and systemic effects. So the local effects, if you get an injection, you're going to have pain, you could have swelling, you could have redness, you could have some of the lymph nodes in that area, draining that area, swelling. Those typical local reactions, itchiness, redness, typical. Then you could have systemic effects, like many people who have children and you've taken your children for vaccination, you know that afterwards they can have fever, they can feel tired, you know. Those are the typical systemic effects. Then there are extremely rare effects, like we saw for the COVID vaccines, where you're observing one effect in several millions. For example, myocarditis was a side effect of these mRNA vaccines, but it was rare.
I'm curious about research funding, particularly of the kind that comes from elsewhere, you know, other countries. It's not our own resources being used to fund research. And you remember what we saw in China with the Wuhan lab was a certain kind of research that either couldn't be done in the US, or maybe for some reason it wasn't done there, but it ended up being done in China. You know, and the research they were doing was related to what they call the you know, gain of function research. I'm trying to find out if there is a possibility that some international research funding,
Right.
and even labs, you know, could be established here, which would in effect be Trojan labs.
Yeah, so gain of function is quite controversial. Yeah, so, but to just briefly explain, gain of function mutations are when, for example, if they're being engineered in the lab, is when, for example, you take a pathogen or a virus and then you alter its characteristics either to enhance its transmission, so it's how infectious it is, or you want to change how pathogenic it is, like how much harm it can cause to a person. And some of the reasons why we would do that, or why it is done, is to create a solution for it. You want to create new treatments. It's like you're trying to get ahead of the problem. So you try and predict what are the possible mutations that will happen. So the problem is that obviously it can be used with for bioterrorism and, you know, with nefarious intent, and that's why it's controversial. Most people don't like it. But as far as locally goes, the kind of biosafety labs, you know, labs have different levels of safety, and the more infectious a pathogen is, like the kind of research when you're doing for Ebola virus, the kind of lab infrastructure you need and protection, personal gear, everything, the ventilation systems are quite advanced compared to just if you're studying malaria, for example. Those kind of facilities, I am not aware that we have them here. So it is quite unlikely for us to be conducting gain of function research here. And for many other reasons, we are unlikely to be a good place for somebody to conduct such research because our security systems are not the strongest. So somebody who would want to do that would probably want to do it in a safe way, probably with a lot of confidentiality. And I don't see like we have reached that level where we have that kind of infrastructure.
So part of the reason that we are not funding research, we are not doing things, sometimes the right way, has to do with our culture of doing things. Mm-hmm. And I wanted you to explain the role of culture in getting ourselves in the situation that we are in, a situation of dependency.
Right. So for me, that's a very important question, because one of the reasons why we are so behind in our research is and let me give you a story. So I was listening to a public health physician from Finland, and she was explaining how she actually acquired COVID. She was one of the first cases in her country to get COVID. And, it was terrible for her and everything, but she donated her blood, her serum, to develop diagnostics because there were no existing tests to, diagnose this disease. But if you come back to our local situation here, and we look at patient zero or just the initial cases who got COVID, it was like they got some kind of incurable. There was a lot of stigma associated with COVID. The way their bodies were buried created a lot of stigma associated with COVID. And generally, even in my medical practice, when I was practicing, there's a lot of superstition around things that really we have no business being superstitious about, because this is just a solution to a problem. But even around HIV, there's a lot of stigma that is not necessary. It is sexually transmitted, but at the end of the day, it's a disease that needs treatment like any other. So this thing where there's a lot of judgment and stigma associated with these diseases interferes with research, because that was an opportunity. All countries were the same when COVID broke out. There were no drugs, there were no diagnostics, there were no vaccines. So that was an opportunity for us to study it locally and discover how it works here. What can we also develop the same things that were developed outside? Obviously, they have infrastructure, and we know another pandemic is going to come just because history repeats itself. These are cycles that have happened in the past. Bubonic plague, smallpox. There's always been pandemics. There will continue to be pandemics. So given that we have that understanding, shouldn't we at least invest in some kind of infrastructure? Not only for care and treatment, but also to do research in such settings. And that requires us to change our mindset, because research is a long game. It is not something that you're going to just pounce on and today you get all your benefits or you cash in. So we need to develop a long view of research, and we also need to minimize our superstitiousness. Some things you just need to step back and deconstruct it, understand how it came to be, put the pieces apart without having a lot of judgment and sentimentality about it, so that you can solve a problem for the public.
Let's get a little morbid here.
Yeah.
How can we encourage cadaver donations and organ donations?
Right. Again, that's another issue with a lot of superstition, where if somebody has died and probably they had good kidneys, people are not really willing for their bodies to be touched at all. And I mean, I understand. But if you knew the kind of difference that it would make in another person's life, after all, you are not there. And you're not going to use that body again. It should be a possibility that we talk about publicly. It should be in our consciousness that actually I can donate my cornea to somebody else, because you're not going to use it. So such things have a lot to do with our own superstitions, our own mindsets. Even cremation, as you mentioned earlier, cremating people is also a very big deal. So I know these are sensitive topics because we are a very spiritual nation, but it is harming us in some ways, these mindsets, because sometimes you just need to solve a problem and you need to think about how to go about it.
But if I donate my eyes, how do I get to see St. Peter?
You might be surprised, you'll still see him anyway. You might be totally surprised. You may not need, those are not the eyes that you're going to use there. So,
okay.
Yeah.
Now, there is the issue of regulation. I know the Pharmacy and Poisons Board is involved as one of the regulators. Could you talk about the others regulatory bodies that ensure our health safety?
Yeah, so like I was saying, we have this Pharmacy and Poisons Board to approve drugs that are approved, for example, in other countries, but they also approve the research. Then we also have ethical committees, which I had mentioned before. So ethics committees, they look at there are certain aspects there are certain conventions around the world that are necessary anywhere you're doing research, whether you're in Brazil or you're in Australia, those rules apply. That research needs to respect people's autonomy, that they can understand what you're saying and then make an independent decision, independent of you as the researcher, whether they want to be involved with it or not. And also that research needs to have a clear benefit attached to it. It needs to have a good clearly attached to it. So there are these ethics committees, including NACOSTI in Kenya, that oversees all the ethical committees to make sure that they are conducting research in a way that is ethically responsible. So you can't just, as a researcher, just be driven by data acquisition and not considering the humanity of other people. And that is also something that we need to inculcate into students at university, that the ethics of whatever you're doing, there needs to be a clear benefit of what you're doing to society. And again, that's a discourse that we are not having. We tend to just skim through things and deal with them on a very shallow level, but this is something that should be in the consciousness of university students. Your creativity has limits. Your innovations have limits. They should not just be like the Nazi war experiments where you're just doing it to gain knowledge. There needs to be a good attached to it.
Does KEMRI have a regulatory role?
All they do is conduct research, but they have an ethics committee to oversee the kind of research that they do.
Yes, I wanted to go back to the issue of our health status, and particularly the apparent rise in cancer cases,
right,
and other non-communicable diseases. So could you tell us what is really going on from a medical perspective?
Yeah, so there's actually what is called a demographic shift that is happening, where people are actually living longer thanks to vaccines. And I just like to point that out because many people oppose vaccines, but the increases in lifespan that we have seen are definitely attributable to vaccines. Water and sanitation and vaccines are key components in the advances in longevity. So because of that, we are seeing a lower infant mortality and longer lifespans. Of course, treatments, advances in technology. If you have cancer, it's not a death sentence in many places because of treatments and all that. So the rise in cases are definitely related to lifestyle. We are much more sedentary than before. There are lifts, there's motorized movement. We are not walking as much or running as much. You can get into a car, you can get into a taxi. Our diet has also changed a lot to become very calorie dense. So typically, they compare this current lifestyle to the previous lifestyle of a hunter-gatherer. And where those communities still exist, for example, in some parts of Tanzania, in the past they've researched them, and they realized you're probably eating 1 meal in a day, whereas today I'm probably eating 6 times in a day, and snacking in between. So all those lifestyle factors are affecting our bodies. Definitely stress. Even though the quality of our lives has seemingly improved, the stress levels are definitely higher because we can see much more mental illness. And that's part of the contribution of the non-communicable disease. We are also seeing a lot of chemicals and toxins, plastics and all these microplastics. And all these things, pesticides, they are probably contributing to the rise in cases that we are seeing.
You wrote your PhD thesis on TB vaccines, and the choice of trial sites. So what was that all about?
TB is a big problem, not all over the world, but here in Kenya it's a big problem. It had actually been subsiding, but once the HIV pandemic came up and there was suppression of the immune systems, then we saw a resurgence of TB again, and it remains a big problem. Now for TB there are vaccines, which is BCG. The problem with BCG is that even though it works very well in the northern hemisphere, in higher latitudes, it actually doesn't work here because of the kind of cases we see. Even among people who don't have HIV, there's still a very high prevalence of HIV and of TB. And that just goes to show you that you need to develop your own in-house solutions, because BCG in the UK works 80%, it's efficacious, but here even 20% or less. So that study was to prepare, the study for my thesis was to prepare for new TB vaccines. And research requires certain infrastructure. You need labs, you need trial sites to collect samples, because pediatric TB is quite different from adult TB. Adults can cough, but children don't have the developed chest musculature to cough enough to expectorate sputum. So you need some specialized facilities to force that sputum to come out, and in the process you generate these aerosols that are potentially harmful. So you need a specialized room with certain air extraction methods to make sure that you're not harming the people around, you're not generating TB that is floating around in the air and infecting people. So all that infrastructure needs to be developed before you can do a TB vaccine trial, because the point of a TB vaccine trial is to compare the number of cases between people who got the vaccine and who didn't get the vaccine. So that thesis was about looking at all those factors that need to come together for us to have a new TB vaccine to bring down the new number of new cases and the number of deaths due to TB.
I wanted you to explain who a pulmonologist is. And my own understanding is that something to do with the lungs, but you obviously can be able to delineate that area better than I. And I wanted you to also give us a status of the African lungs, given the increasing pollution, the delicate nature of the lungs, as we saw during COVID. This is one organ that I never thought was important, but turns out to be extremely important.
Critical,
And also given the propensity of biological warfare adherents to basically build diseases that affect the lungs.
So to start off, I'm not a pulmonologist. I worked in respiratory diseases like TB for some time, and in that process you interface with other diseases of the lung, but I'm not a pulmonologist. I can still speak to some of those issues because they are related. So the lungs, if you look at the surface area, I forget exactly how big it is, but it's a very vast surface area, and with every breath you take, you end up, at the end of the day, you're taking in thousands of liters of air. So the quality of the air, the particulate matter in it, and and the amount of oxygen that is in it matters a lot. That varies with altitude, for example. So as we live in cities and densely populated areas, like in Kenya, sometimes there's no proper zoning between industrial areas and residential areas. We're exposed even to a lot of pollution. Even aside from industrial pollutants, there's just the diesel and the vehicles, there's a lot of pollution. And with this vast surface area, if you were to actually take a lung and spread it out, it's a very large surface area. So there's a lot of interfacing between you and your environment. And so that's why it's so easy for people to catch respiratory diseases, including diseases related to pollution. Cancers are related to pollution. Chronic lung disease like emphysema and chronic bronchitis are related to pollution, and we are seeing the incidences on the rise for all these diseases. So again, it comes down to our lifestyle. So typically a pulmonologist is going to handle all those issues, including reactive airway disease like asthma, and also rarer diseases. There are also some rare inherited genetic diseases which are also happening, like cystic fibrosis and all that. So that's the typical field of pulmonologist. But for me, I am a clinical trialist.
What needs to be done to ensure utmost safety, health, because this is all about human safety?
Okay. Yeah. First of all, I would like to say that Kenyans have some things going for them as far as research goes. Number one, our disadvantage in terms of poverty and lack of resources is an opportunity for us to innovate. Necessity is the mother of invention. We should not lose sight of that. Number two, this is a country with a highly educated population. There are a lot of smart people in every field, not just in research. In every field, there are innovators, there are people who have deep understanding. The expertise is here. And without that human resource base, you cannot grow any country. That's a big plus that we have. We also a growing population. That's a big plus. There are countries where the population is on decline, but we are a growing population. That's a big plus. So there's a market for the product, and there's also a source of innovation for the product. So those are big things going for us that we have not tapped into adequately or talked about. And those are big advantages for research. Number two, we already have a basic research infrastructure. We have universities, we have medical research institutes, and so on and so forth. What is not happening is the synergies. So people are not coming together enough. And when we come together, we don't have meaningful discussions. We either attack each other or attack leaders and focus too much on politicians when the people themselves have power. So just coming together as individuals has power to solve problems without a politician being a part of the equation. And we've seen that before. We saw it with Gen Zs. We've seen it with, like, for example, the Equity Bank and different other innovations. It's not only, everything is not solved by the politician. Yeah, that's a big point for us to think about. And we would like media, like, I find what you're doing very meaningful, because it's forcing us to think about other things other than just hype and noise and chaos and the politics of the day. We actually have solutions just by getting together. So that would be good if we can begin to have more discussions like this and make them more mainstream and cutting across sectors. So in the media, in banking, in research, in agriculture, we need to talk about this. I love that discussion about mining, because a lot of the issues that came up are the same issues that are faced in research. So if anybody has not watched that one, I recommend that they go and watch, because there's a lot of overlap in the issues, including over-reliance on politics. You can still form partnerships without politicians. They are important and they help, but we should not always think that they are the silver bullet. Finally, I want to say that it's important for us to believe in ourselves. We actually have a lot of things going for us. You don't need to have to be born with a silver spoon in your mouth to make it, so to speak. So as a nation, we have a lot of wealth, as we saw in that interview with the discussion with the mineral wealth. But even the things that you don't have, chances are there's an advantage in that, like I mentioned with the cases in India and everything. So we need to believe in ourselves more as Kenyans and stop bashing. There's too much bashing of ourselves, our resources, our abilities, where we are versus other countries. And that interferes with your innovation. It interferes with your psyche. It makes you think that you need to go somewhere to do well, while the things you need, you probably have enough to get started with right here. When it comes to the status of our health, I would say we need to think as a group. We Kenyans, we are not good at thinking as a unit. So for me, I would think about myself and my family. If I have insurance or I can pay my bills, then I would not, the rest I leave to politicians, or I leave it's someone else's problem. But the more we think as a unit, the stronger we are. The more I think, I can solve this problem for myself, but I'd be better off by coming and talking to you, Prof. It elevates us as a people. So we need to break down the silos of our individualism. If you do well by yourself, you haven't done well. Your children are still going to have to deal with the problems because your neighbor has not done well. It's not good for you. Come together with other people. That is where your strength is. And the more siloed we are, even as Kenyans, that's why politicians can take advantage of us. That's why other nations can take advantage of us. The more we think as a group, like, how can we as Kenyans do this? How can we as the media practitioners do this? The greater your voice is, the greater your impact. When you look at teachers, one of their greatest strengths is the fact that they usually come together and speak as a unit. And if you look through time, the people who have gotten the greatest increments, salary increments over time, is a teacher, because they may not be the most wealthy as individuals, but when it comes to coming together, they don't joke. So we need to copy that and replicate it in every sector of this country. The more we come together, the farther this country will go. And the more you think of yourself, the worse it's going to be for you and for your children.